The college comprises 5 schools: agricultural, earth and environmental sciences, engineering, chemistry and physics, life sciences and mathematics, statistics and computer science. Engineering is offered on the Howard College campus, agriculture on the Pietermaritzburg, and all the other sciences on both the Pietermaritzburg and Westville campuses. Exceptions are agricultural engineering, which is offered in Pietermaritzburg, and geology, which is offered at Westville only. To compete with the best and to meet the challenges of the 21st century, South Africa needs highly skilled professionals in the areas of agriculture, engineering, and science. We offer a wide array of interesting and innovative degree programs that open doors to a variety of different careers. Some degrees are unique to UKZN and are not offered at other tertiary institutions. For example, we are the only university in South Africa to offer a fully accredited agricultural engineering degree, and one of only 2 universities in the country to offer a BSc in land surveying. Our postgraduate food security training program is the only one in the world, and we have the widest range of agricultural disciplines at any single South African or African campus. The choices of degree and the options on offer may seem confusing at first. Engineering degrees are more specific, and the options are usually career-focused. Emphasis is placed on training in the specific principles which engineers deal with in their professions. Engineering offers 7 different professional specializations. There are also degrees in land surveying or geomatics and property development. In science, there are structured or career-focused degrees. These include fields as varied as chemical technology, marine biology, industrial and applied biotechnology, and computer science and information technology, to mention a few. There are also more general degrees with a lot of flexibility. In this case, students have the choice of studying a BSc M stream in maths, physics, computer science, and statistics. All of these have a strong mathematical component. The other option is a BSc LES stream with a focus on the life and environmental sciences with subjects such as geography, genetics, botany, hydrology, zoology, ecology, and microbiology. In agriculture, there are various different options that involve practical and scientific applications. Students can combine science subjects with specific agricultural disciplines for a 4-year Bachelor of Science in Agriculture degree. These are more career-orientated. Agriculture and agricultural management are 3-year degrees with a higher practical component. Dietetics is a professional qualification that deals with the role nutrition plays in the promotion of health and the treatment of disease. Most degree programs across the college have field trips and projects that contribute significantly to the curriculum. This ensures that students learn in real-life situations under full field conditions. Many of our lecturers have received Excellence in Teaching awards, are recognized internationally, and are rich in practical experience. Students have access to excellent facilities that rank amongst the best in the world. 300 million rands has recently been invested in new teaching and research facilities, state-of-the-art laboratories, and improved equipment. The 30 million rands new School of Engineering building is the envy of engineering campuses across the country. Agricultural students have access to a 356-hectare research and training farm situated near the Pietermaritzburg campus. This facility provides students with hands-on training and is used extensively as an outdoor laboratory for teaching and research in areas such as animal and poultry science, horticultural science, and agriculture.
Okay, good evening, students, and welcome back to our session on life sciences for your Matric Helpline. And thanks for joining us. It's our final session now. And I hope you guys have all learned something thus far. That's why you're here tonight. And tonight we're going to be doing genetics. It's the last component of your exam. Okay. If you just give me a moment, I'm just going to start sharing my screen shortly. There's some students still joining, so I'll give it a minute or two. Okay, students, can you see my screen? Just give me a thumbs up or just say yes. Okay, can everybody see my screen? Okay, thank you. Okay. Okay, so we're going to get straight into it. We're starting with genetics, okay? And the things you need to know in genetics, in the genetics section, first of all, is your terminology, okay? So you need to know quite a range of words that are specific to this section, okay? Let me give you a few examples. Things like homozygous, heterozygous, what is an allele, what is a genotype, a phenotype. Okay, so what is a karyotype? Okay, so that type of terminology, dominance, codominance, okay, you must know those definitions because they will definitely come up in your give a biological term or multiple choice, or you know, there's this other question that comes up in your paper. It says select A or B or both. Okay, and it's usually like in a little table. So that usually deals with your definitions in genetics. Okay, so please make sure you know words like segregation, recessive gene, mutation, what is a locus, okay? Things like that you shouldn't be able to, you should know the definitions of. Okay, that is your terminology in the genetics section. Next, you need to know about mono and dihybrid crosses. Okay, so a monohybrid cross is when you're only looking at one characteristic, and a dihybrid cross is when you're looking at two different characteristics, okay, on the same individual. Okay, and then multiple alleles, okay, that is when you're looking at, for example, blood types, or you're looking at more than one allele for a specific characteristic, okay? Then your sex-linked disorders, okay, that is your male or female inherited disorders, okay? And then genetic engineering, okay, that is like your genetically modified foods, how we can use genetics to improve our economy, and how it's used in industry. Okay, so that is genetic engineering. Okay, and you also need to know, I just need to add it in here. You also need to know pedigree diagrams. Sorry. Okay. So that is another thing that you need to be able to read off from and do. Okay. Okay, so let's start with our biological terms. Give the biological term for the alternative form of a gene on corresponding positions of homologous chromosomes. What is that called?
Fanelo says alleles.
Yes, that is correct. Okay. And what is a gene that is not expressed in the phenotype unless found in the homozygous state? Can I have an answer? A gene that is not expressed in the phenotype unless it's in the homozygous state.
Recessive allele.
Yes, correct. A recessive gene. Okay, what is different alleles of a gene on a chromosome? When you have different alleles of a gene on a chromosome. Anybody want to try? That is, it's an easy one. Different alleles of a gene on a chromosome is called a heterozygous state. Okay, you got two different alleles, a recessive and a dominant. So heterozygous. Okay, what is the change in the structure of a gene or chromosome?
Mutation.
Yes. Chromosomes of similar size and structure and contain the same genes. We did this earlier today in the morning.
Homologous chromosome.
Correct. Okay. Okay, so let's start with our short answer questions. Okay. Just to get us going. In rats, black fur is produced by the allele, big B, and white fur, small b. The table shows the genotypes of some rats. Okay. What is the phenotype produced by the recessive allele? So which is dominant, black fur or white fur?
Black fur.
Correct. Okay, so what is the phenotype produced by the recessive allele?
Santuri says black fur is dominant.
Yes.
If black is dominant, what is recessive? Santuri said 3.
No, the question is, what is the phenotype produced by the recessive allele?
So Santuri said white fur.
Correct.
That's funny.
Correct. And now what is the phenotype of rat 2?
Black.
Correct. Give the number of the rat that is or are purebred.
Rat 1 and rat 3.
Correct. Homozygous dominant.
Rats 1.
Correct. Okay, that was an easy one just to get you going, get your blood flowing. Okay, next. Okay, if we look at this pedigree diagram, we're looking at one regarding hemophilia. So females with hemophilia, males with and without hemophilia. Okay, so we've got two people, a female without hemophilia and a male with hemophilia, and they have children. Okay, two have hemophilia and two do not have hemophilia. Okay, which one of the combinations in the table below is correct for both individuals 1 and 5? So 1 is the female without hemophilia and 5 is the male with hemophilia. Okay, so look at that table there. Phenotype of individual 1. The phenotype of individual 1 is a female without hemophilia. Okay, so that is either A, B, or D. Okay, and now number 5 is a male with hemophilia. Okay, so look at that genotype there.
Santuri said D.
Okay. Any other takers?
User says C.
Okay, so hemophilia now is your sex-linked characteristic, okay? And it's found on the X-axis, X chromosome. And its gene is represented by a small h, okay? So it's always a small h on the X. Okay.
Fanello says B.
Oh, everybody's giving me different answers. Okay. So the condition shows only in the recessive state in females. Okay. But if it's on a male chromosome, then the male will be X small h y. Okay. That is a male with hemophilia. Okay. So where do we have X small h y? The answer is?
Fanello said B.
That is correct. Okay, so B. So you only have, if the female has to show be a hemophiliac, it will be X small h, X small h. If the male has to have hemophilia, it'll be X small h, Y. Okay. If the female is a carrier, then it will be X big H, X small h. Okay, if she's a female without hemophilia but is a carrier. Okay. Okay, so what is the percentage chance that individuals 3 and 4 have a child with hemophilia? So you have a female with hemophilia and a male with hemophilia. What are the chances? Anybody want to give it a try?
User said C. 25%.
C25. Any other people got any other answers? Okay, that is correct. Okay, it'll be 25% because each parent has a 50% chance of having a child with hemophilia. Okay. Okay. In dogs, coat appearance is controlled by 2 genes. One for coat color and one for coat pattern, which is the presence or absence of white patches. The alleles for each characteristic are shown in the table below. Okay, coat color, you have black, which is big B, and gray, a small b. Coat pattern is without white patches, big T, and with white patches, small t. In a cross between 2 dogs, the 4 offspring, V, W, X, and Y, had the phenotypes as shown in the diagram below. Okay, so we've got full black, okay, black with white patches, full gray, and gray with white patches. Okay, what is the term given to a genetic cross involving, excuse me, two characteristics? Anybody?
Dihybrid cross.
Very easy. Yes, that's correct. The dihybrid cross. Okay, give the phenotype of the dominant coat color. Which is dominant, gray or black?
Vanillo and Santuri say black.
Correct, because it's big B. Okay. What give the phenotype of dog V? The phenotype is black. Okay. The phenotype of a dog that is homozygous recessive for both characteristics. So we are looking for a dog that is gray with white patches because that is the recessive. Can I ask that you mute your mics, please? Okay. Please mute yourselves. Okay, so a dog that is homozygous recessive for both characteristics. Okay, so the recessive characteristics are gray and white patches. Okay, so which dog is gray with white patches?
Santuri and Vanillo say Y.
Correct. Okay. Explain why all dogs with the phenotype of dog W may not have the same genotype. So we're looking at a black dog with white patches. Why? It may have the same phenotype, but it may not necessarily have the same genotype. And why is that? Anybody? Because black with white without white patches is both dominant. So they will have the same phenotype, but they may not necessarily be the same genotype because you can be homozygous dominant or heterozygous dominant. Okay, and that will both give you black without white patches. Okay. Okay, yes. The two characteristics are inherited in accordance with Mendel's principle of independent assortment. State the principle. Okay, what is Mendel's principle of independent assortment?
Seema said co-dominance.
No. You need to state the principle. What is the principle of codominant? The principle of independent assortment. State the principle of independent assortment. Okay, it is, write this down. Alleles of a gene for one characteristic segregate independently of the alleles of a gene for another characteristic. Okay, so alleles of genes segregate independently from each other. That's what that means. So each gene is separating on its own, independent of other genes. Okay, then the alleles of each gene will therefore come together randomly during gamete formation. And that is the law of independent assortment. Okay. Okay, read the extract below. Stem cell surgery has been performed for the first time in South Africa at a Cape Town hospital. A patient became paralyzed in a driving accident. He had no movement or feeling in any of his limbs because his nerve cells were damaged. Embryonic stem cells were used to attempt to correct a defect in the spinal cord of the patient. He has now developed partial sensation throughout the body. Explain why stem cells are suitable cells to use for the treatment of this patient. For 3 marks. Okay, what is special about stem cells? Nobody's talking to me tonight. I need some answers.
The stem cells can form into any tissue or organ. Therefore, they can be used to replace damaged cells.
Very good, that is correct. Okay, so stem cells can be made into any, they have the potential to become any kind of cell. Okay, embryonic stem cells are extremely what we call pluripotent, which means that it's able to become any type of cell. Okay, and that they're also undifferentiated, which means they still they're not assigned to any cell type as yet. Okay, so it can become any cell type. Okay, stem cells have the potential to develop into any cell type and also to replace the nerve cells that are damaged. Okay, so it can become nerve cells. Okay, an embryo is a potential life, which means it could develop into a baby, or it possesses cells that can develop into any tissue. Okay. Okay, explain why some people prefer the use of umbilical cords as a source of stem cells rather than the use of human embryos. Okay, an embryo or an umbilical cord. Okay, there's obviously moral and ethical issues, okay, around using an embryo, okay, because that is obviously formed, has the potential to become a baby. Okay, and umbilical cords are usually discarded, so it's easy to use the umbilical cords, and they do not pose moral or ethical issues. Okay. Okay, a baby was kidnapped from a hospital immediately after she was born. 15 years later, it was discovered that Mr. and Mrs. Thomas, who were raising her, were not her biological parents. Mr. and Mrs. George, whose baby was born around the same time, claimed that she was their child. The blood groups of both families are shown in the table below. Okay, so we got the child, which is blood group O, and then Mr. and Mrs. Thomas and Mr. and Mrs. George. Okay, so how many genes control the inheritance of blood groups? How many genes?
Santuri said 3.
Okay, so it's asking you how many genes, not how many alleles. Okay, so there's 3 alleles, but 1 gene. Okay, so 1 gene, but 3 different alleles. So that's why blood group types are referred to as multiple alleles. Okay, name the individual whose blood group shows co-dominance.
Mrs. Thomas.
Correct, because it's both A and B. Explain why Mr. and Mrs. George could possibly be the parents of this child. How could they possibly be? If they were, if Mr. and Mrs. George were heterozygous. If they were heterozygous, then they would be able to be the parents of the child if they were both heterozygous. Okay. Okay, polycystic kidney disease, PKD, in dogs is caused by a dominant allele H. The pedigree diagram below shows the offspring produced by 2 dogs, 1 and 2. Okay, a male without PKD is a blank square and a male with the PKD is a black square, and females are the same. Okay, so how many offspring are affected by PKD? How many offspring?
Two.
Yes. Explain how the genotype of offspring A shows that both parents are heterozygous. What tells us that both parents are heterozygous? The fact that we have offspring A. Okay, offspring A is a male without PKD. So for him to not have PKD, it means that he had to receive a recessive allele from both parents, which means that the genotype of both parents is heterozygous dominant. Okay, are you with me? Are you with me? Hello? Everybody
Yes, ma'am.
understand? Okay. Is everybody understanding so far? You please stop me at any point if you don't understand. Okay. Okay. And read the following extract on artificial selection. Broccoli, Brussels sprouts, and cauliflower are edible plants in the cabbage family. They are different varieties of the same species. Okay, they produce a cluster of yellow or white flowers. Each flower has 4 petals, 4 sepals, 6 anthers, and a single stigma, style, and ovary. The somatic cells of these plants have a diploid chromosome number of 18. The chromosome number is reduced from diploid to haploid during gamete production, specifically during anaphase 1 of meiosis. The size of the cabbage head was always important in artificial selection projects, but now the focus is on increasing resistance to various insects and diseases, and on improving the nutritional content of cabbage. Define a species. What is a species? Anybody? Students, you need to give me some answers here. Anything on the chat?
No answers on the chat. Fanelo says any living organisms.
Okay, be a little bit more specific.
Yeah.
What about these living organisms? What are they able to do?
Reproduce.
Yes, very good. Okay, so it needs to be an organism that is able to reproduce and interbreed and produce fertile offspring. Okay. Name 2 structures in a flower where meiosis takes place. Where does meiosis take place? In a flower.
Fanelo says the anther and S says ovary.
Very good. Okay, that is correct. Okay. The anther and the ovary. Okay, now how many of each of the following structures is present in each cell of Brassica in the process of meiosis? Okay, so they've told you the diploid chromosome number of 18. Okay. So it's now asking you how many of each of the following structures is present in each cell. It's chromosomes at the end of telophase 2. Students, chromosomes, how many chromosomes at the end of telophase 2? You started off with 18 chromosomes.
9 chromosomes.
Correct. Okay. And the number of chromatids during metaphase 1. Just try.
Students are saying 4.
4 chromatids.
Remember, you have 18 chromosomes. So the chromatids at metaphase 1 will be.
Okay, so remember it has not split into each cell yet. Okay, it's still in metaphase 1, which means you're going to have how many chromatids? If you've got 18 chromosomes, how many chromatids do you have?
Yeah. 36 chromatids.
Yes, very good. Thank you. Describe the events of anaphase 2. What happens in anaphase 2? Can you see now they are they're mixing the genetics section with some of the DNA section. Okay, so you will get questions like this where things are a bit mixed together in different questions. Okay, so describe the events of anaphase 2. What can you tell me happens in anaphase 2? You need to know this. This should be very easy to you. Are there any answers on the chat?
No answers. And those, okay, so S said chromatids pull to opposite poles at NFAs 2.
Okay, yes, that is correct. Okay, but it's saying describe the events. So don't just say chromatids pull to the opposite poles. You need to say that. You need to give some more detail.
Okay, so Fanelo says the chromosomes split and the chromatids are pulled towards the opposite poles.
Okay, and what did the chromatids look like? Remember, they've undergone crossing over, so they are combined. Okay, so you can say recombined chromatids, or you can talk about the number of chromatids. Okay, but you got to say something for 3 marks. So you can say the spindle fibers contract. Okay, and the centromere of each chromosome splits into 2, and the chromatids move to the opposite poles. That is actually the full answer. Okay.
Please, can you repeat the answer, ma'am?
Sure. Okay, it is the spindle fibers contract. The centromere of each chromosome splits into 2. And the chromatids move to the opposite poles. Everybody got that?
Yes, thank you.
Okay. Explain why the focus is now on increasing resistance to insects and diseases. Why do you think there is attention focused on increasing resistance to insects and diseases? So basically, you're genetic engineering. You're trying to modify the plant to be resistant to these insects and diseases. What is the purpose of that? For food security, okay, there'll be fewer crops that will be destroyed by diseases and insects because they are now resistant to it. So genetic engineering will actually make that plant resistant to insects and diseases. Okay, and that will then increase the crop yield, increase food security, and decrease the expenses on pesticide. Okay. A species of fish has 3 phenotypes for fin length: elongated, short, and medium. Heterozygous fish have medium fins. Okay, the characteristic under the control of one gene with 2 alleles, elongated and short. Name and describe the type of dominance shown here. What type of dominance is this?
Incomplete dominance.
Why would you say that? Because neither the elongated nor the short are dominant. You have an intermediate, and that intermediate is your medium. Okay, so that's how we know that it is incomplete dominance. Okay. Use a genetic cross to show the percentage chance of 2 fish with medium fins having offspring with short fins. Okay, so you need to be able to do a genetic cross. Okay, and I'm going to switch slides here just going to stop sharing and switch slides so you can see how to do a cross. Can you see my screen?
Yes, we can see the screen.
Okay, can you see a monohybrid cross at the top there? I just want to see which screen you are seeing.
You're seeing the monohybrid with the Punnett Square,
Okay.
but it's
Okay.
minimized. I can see your desktop as well.
Oh, okay. Okay, now?
Now it is the PowerPoint.
All right. Okay, so when you're doing a cross, you need to always make sure that you are writing the genotype of the parents. Okay. Okay, first you need to decide, okay, what is dominant and what is recessive. Okay, so that is your first step in doing your genetic cross. Okay, so you can write these steps down so that you always doing the correct thing. Okay. Okay, so first, when you're doing a monohybrid cross, you're going to decide whether the genetic cross is incomplete dominance or complete dominance. Okay. So in our case, we've said it is incomplete. So choose the symbols that you're going to use. And now they've told us, okay. Oh, I can't go back to that slide now. They've told us that the characteristic under the control of one gene with two alleles, elongated and short. Okay, so they've told us those are your symbols that we're going to use. Okay, and we've said it is incomplete dominance. Okay. Okay, so just as this, okay, they've said let big B equal brown eyes and small b equal green eyes. Okay, you must write it out like that. Okay, and then you must then write the phenotype of or the genotype of the parents. Okay, so the mother's genotype is small b, and the father's genotype is big B, small b. Okay, so you must write it out like this, okay, to get the marks. And then you need to write the gametes. Okay, so you can see a big B, small b, small b, small b from each of the parents. Okay, so those are the gametes. And now those gametes can all have a chance of combining with each other. Okay, so there is when we now do our Punnett Square. We put our father's genes, big B, small b on the top, and our mother's genes, small b along the side. And each of those gametes can now pair together. So big B can pair with small b, and that goes in the block. Small b can compare with small b, and that goes in the block. And again, big B and small b goes in the block. Small b goes in the last block. Okay, so each gamete can pair with each and every other gamete. Okay, so that is when you do your Punnett Square. And then step 7 is now actually, sorry, step 8 is now actually writing the genotype and phenotype of that F1 generation. Okay, so now you're going to look at your Punnett Square and you're going to see, okay, what does that tell us? The first block is big B, small b. What is the genotype? Okay, let's count how many we have that is of the same genotype. we got 2 big B, small b's. Okay, so our genotype is 50% big B, small b, and 50% small b, small b. Okay, now the corresponding genotype of the corresponding phenotype of that is 50% are brown-eyed and 50% are green-eyed. Okay, so you must always, after you do the Punnett Square, you're writing your genotype and your phenotype. So let's go over that one more time just so you know the steps. Okay, so first you need to decide whether the genetic cross involves complete or incomplete dominance. Then choose the symbols that you're going to use. Then write down the phenotype of the 2 parents. Okay, then write down the genotype of the parents. Then show the gametes of each parent. And then you're going to cross each gamete or multiply each gamete. Okay, and that you do in a Punnett Square. Okay. So each gamete has a chance of pairing with each and every other gamete. Then, once you've completed your Punnett Square, you then write your genotype and your phenotype from the Punnett Square. And we said to you earlier that you should always use highlighters or use some way of being able to count all these genotypes and phenotypes. Okay, so it's not to make a mistake. You rather use different colored highlighters or pens. Okay. Does everybody understand that? Any responses? Do you understand that? That is the format that you must always do for a cross. Okay, are you with me still?
Yes.
Okay. Okay, so how are we going to do the cross with 2 fish that have medium fins? What are the chances of them having offspring with short fins? Okay, so cross 2 medium fins. What will be our medium fin? Medium fin will be ES. Okay. Okay, so we've got our medium fins, ES. So our phenotype of the both parents are medium fins. Our genotype of the parents, both are ES. Then we do our Punnett Square. We're going to put E and put S in a table, horizontal and vertically. You're going to write E and S. And now you're going to do your cross. So you end up with EE, ES, then ES again, and then SS. So now you're going to count all of those offspring in the Punnett Square, and you get 1:2:1. Okay, you get one, what do you call this? One elongated fin, homozygous dominant, 2 medium fins, okay, because it's both E, so it's ES, and then 1 SS. So that's 25% elongated fins. 25% short fins. Okay, so the question asks, what use a genetic cross to show the percentage chance. Okay, so from that Punnett Square, what's the percentage chance of them having short finned offspring? And that is only SS. Okay, so what is the chance? It's a 1 out of 4, which is 25%. Okay.
Ma'am, what type of dominance is this?
What type of dominance?
Yes.
We said it was incomplete dominance.
Okay,
Because
thank
you have an intermediate. Okay, elongated and short, and you have a medium. Okay, if there's a intermediate, then it means it's incomplete. So remember, incomplete, intermediate. So it's both starting with the in. Incomplete, intermediate. Remember that. Okay. Any other questions? Are you okay with this genetic cross? You must be comfortable with doing one of these. Are you with me, everybody? Yes or no?
Panelo said yes.
Okay. Okay, then in holly trees, red fruit is dominant over white fruit. Okay, red fruit is dominant over white fruit, so it's using big R and small r, and spiny leaves is dominant over smooth leaves. So that's another characteristic. Fruit color and type of leaves. Okay, spiny leaves is dominant over smooth leaves. Okay, big L, small l. The Punnett square below shows the possible results of a cross between two individual parents. The genotype at X is not given. Okay, so you can see the gametes there of the parents. Now each parent has an allele for each characteristic. Okay, you can see R and L there. Okay, so what type of dominance is this? What type of dominance? Hello?
Yes, said complete dominance.
Complete dominance. And what type of cross is it?
How many traits are we looking at?
Dihybrid cross.
It's a dihybrid, correct. Okay, give the genotype of X. Okay, so you're going to say RL crossed with RL. It's going to give us a big R, big R, big L, big L. Okay, and now what is the phenotype of the parents? Okay, what's the phenotype of the parents?
So look at the gametes on the left-hand side and on the top and group the fruit color together and group the leaves appearance pattern together, and you'll get your genotype, and then from there you can derive the phenotype of the parents.
Yes. Okay, so what type of fruit?
Okay, so the parents on the table, so where you have gametes, the first row on the top next to gametes, that's one parent, and the column below gametes, that's the as second parent.
Okay. Okay, so our parents are, the genotype is big R,
So-
small r. Yes, sorry.
S, I said it's red fruit with spiny leaves.
Okay, yes, that's correct, because we have the genotype of the parents is big R, small r, big L, small l for both parents. Okay, and that translates into red fruit with spiny leaves. Okay, in a population 128 plants, in a population of 128 plants, how many plants with red fruit and smooth leaves are expected from the Punnett Square above? Show all your working. Okay, so what is the genotype for red fruit and smooth leaves? It will be the genotype would be for red fruit and smooth leaves. So how many plants have red fruit and smooth leaves in this Punnett Square? Students waiting for some answers.
And Fanello says 4. And S says 3. So let's look at it.
Okay, how many plants with red fruit and smooth leaves? So we're looking for big R and small l, small l. So we've got 1, 2,
3 and S says
3,
3.
4. Okay, look for the ones with, oh, sorry, you're right. There's 3. Okay, not 4. Okay, the fourth one is actually white fruit. Okay, so we've got 3 that are red fruit with smooth leaves. Okay, so 3 out, and now in a population of 128, how many would that be? So 3 out of 16, and then you need to be able, now you need to times that by 28. So 3 out of 16 times 128, do that in your calculator, and you're going to get 3 over 16 times 128, 24, correct. Okay. A farmer wanted to produce plants with only white fruit and spiny leaves. Okay, give the genotype of the plants that he should use in the cross. Okay, so now he wants the offspring to be white fruit and spiny leaves. Okay, so what's white fruit? Small r, and spiny leaves, big L. Okay, so white fruit will have to be small r, small r. Okay, so both the parents need to have the recessive allele. Okay, so both parents, if he wants, see it says here, he wants only white fruit, which means the parents have to be white fruit because that's the recessive allele. Okay, so the parents must be small r, Okay, and then spiny leaves, they can have a big L. So it can be homozygous or heterozygous dominant. So it'll be small r, big L, small l, or small r, big L, big L. Okay, it will also give you spiny leaves. Okay. So those are the parents that he should cross in order to get only white flowers, only white fruit and spiny leaves. Okay, is everybody okay with that? Do you understand? Yes or no? Yes, okay. Okay. Now, the diagram below represents a pair of homologous chromosomes in a plant cell. The allele for two characteristics, seed color and plant height, are indicated in the chromosomes, are indicated on the chromosomes. Okay. Give the term used to describe the position of an allele on a chromosome.
Locus.
Correct, the locus. Okay. Identify parts A and C. Identify parts A and C. What's A?
Fanello says A is a chromatid. And S says C is the centromere.
Correct. C is the centromere. Okay, and A is a chromatid. Correct. Okay. Now, name the process during which parts A and B exchange genetic material.
Crossing over.
Yes. Okay. During which phase of meiosis does the process named in question 1.4.3 take place? So which phase? When does crossing
Metaphase
over
one.
occur? When does crossing over occur?
Metaphase one.
Prophase one.
Not metaphase.
Prophase one.
Yes, that is correct. Okay. During, state the following for this plant. What is the genotype for this plant? A big G and a small g, and a small t and a small t. So what's the genotype? Well, I just told you the answer. It is big G, small t, small t. Okay. Then the characteristic that is homozygous recessive. So which of those things is homozygous recessive?
So S
Plant.
says the height.
Yes, okay, so plant height is the characteristic that is homozygous recessive because it is small t. Okay. Okay, the table below shows the blood groups of the members of a family. Two of the children are biological offspring of the parents and one child is adopted. How many different phenotypes for blood groups appear in this family? How many different phenotypes? Just count there.
S says 4.
Yes, that is correct. Okay. Possible genotypes. How many possible genotypes are there for the blood group AB?
S said 1.
Yes, that's correct. Then give the genotype of the father. What's the genotype of the father? You need to look at the mother and the daughter as well.
So S said I with a capital A and then a small I.
And why is that? Why did you get that answer? How did you get that answer? Because the daughter is the same phenotype. Okay, so that means the daughter needs to have the father has to have a recessive in order for the daughter to be also blood group A. So the father is big I, big A, small I. Okay, which member of the family has the genotype 2? Sorry, not 2, small I, small I.
S said son 1.
Correct. Which member of the family has co-dominant alleles?
Mfanelo said the mother.
Yes, that is correct. And which family member is adopted? Which family member is adopted? Remember, the genotype II, it means that each parent has to have the allele small I. So does the mother have the allele small I? No, the mother has IA, IB, and the father has IAI, small I. So son 1 cannot be the biological child of parents that are IAI and IAIB because there's only one allele of small I. Okay. The mother cannot contribute that small I. She doesn't have it. Okay, so son 1 is the adopted son. Is everybody with me there? Yes or no? You okay?
Yeah.
All right. Okay,
Jerusha,
so yes.
just explain why son 2 is not the option of being adopted.
Why son 2 is not an option? Okay. Because what are the possible genotypes of son 2? Tell me the possible genotypes. Son 2 could possibly be IB or IB small I. So is he IB? No, he isn't, because the father cannot contribute IB. The father can only contribute IA or small I. Okay. The mother, she has that IB allele. Okay, so she can contribute IB. So the father obviously contributed small I. So the son 2's genotype is IB small I. So therefore, the mother and father are his parents because the mother has the IB and the father contributed the small I. Okay, so he is heterozygous for that allele for blood group B. Okay, so that's why son 2 could possibly, it is actually a biological child. Okay, is that fine? Do you understand?
User said thanks.
Okay. Okay, now in rabbits, fur color may be black, white, or gray. The inheritance of fur color is controlled by 2 alleles, black fur and white fur. Explain why fur color in rabbits is an example of inheritance with incomplete dominance. Why do we have incomplete? Why is this incomplete dominance? Because you have an?
Yes, said there's an intermediate.
Yes. So
gray.
incomplete dominance, you have an intermediate. Okay, so the intermediate is gray. So it's a mixture of black and white fur. You get gray fur. Okay, so use a genetic cross to show the expected genotypes and phenotypes of offspring when a gray male mates with a black female. Okay, so we've got, what's the genotype of a gray male? Gray male will be big W, big B. Okay, cross with a black female will be big B, big B. Okay. Are you with me there? Okay. So we have, okay, let's do our steps. We've already been told it's incomplete dominance. Now we have to say what is our phenotype of the parents, gray and black. Then what is our genotype of the parents, big B, W, and big B, big B. Okay. Now what are our gametes? Okay, it'll be big B, W, and big B, big B. Okay, so those are our gametes. Now in the cross, we're gonna cross big B with big B. We're gonna cross W with big B. We're gonna cross big B with big B again, and W with B again. Okay, so our genotypes of the F1 generation is big B, then another big B, then we've got big B W, then we've got big B W again. So we've got essentially 2 big B and 2 big B W. That's our genotype. Now our phenotype is gray and 2 gray and 2 black. Okay. Okay, let me just write that, or maybe, yeah, Nimisha has put that in the chat for you. Okay, so we've got 2 big B, 2 big BW, and that is 2 gray and 2 black. Okay, so that is how you're going to get your 6 marks. Okay. Please, if you're uncomfortable with this, please tell me now. You got to be able to do this for 6 marks. Okay. Let me just put up the model answer to show you what it's supposed to look like. Okay. Okay, you can just have a look at this. Okay, so you can see the phenotype of the parents, the genotype of the parents. Okay, can you see that? Hello?
Yes.
Okay, so you've got your genotype of the parents, phenotype of the parents, then your gametes must be written out like that, and then your F1 generation, you must do your Punnett square. After you write down the gametes, you do your Punnett square, okay? And then you write down the F1, you must write F1 there. F1, your genotypes, list them, then your phenotypes, list them. Okay. So you should also see, you get a mark for saying P1 and F1. Okay, so you must say the parents, you must say the offspring. And then you must say meiosis happened between, so after you write the genotype of the parents, you must write the word meiosis, then write your gametes, okay, then write the word fertilization, okay. That's, see if you can look at the bottom there, that is actually, you've got a mark for that. Okay, so this is where your marks are coming from. Okay, scroll further down. Oh, here's it with the Punnett square. Okay, so when you do the Punnett square, you get 1 mark for correct gametes and 1 mark for correct genotypes. Okay, so this is what I want you to write down. Okay, take a screenshot or something so that you know every time you do a cross, this is what it must look like. It must include P1, genotype of the parents, phenotype of the parents, then meiosis, then you must list the gametes. Okay, then you must say the word fertilization, then you say F1. Okay, and you do your Punnett square. Okay, then what is the phenotype of the F1 generation? Okay, you can also list the genotypes of the F1 generation. Okay, so you must do it like this, please, for you to get all the marks. Is everybody following? Are you with me?
Yes, they are.
Okay. Okay, then last question. In humans, the ability to taste a certain substance is inherited and is controlled by the dominant allele big T. People who are able to taste the substance are called tasters, while those who cannot are called non-tasters. The pedigree diagram shows the inheritance of this trait in a family. Okay, so we've got a male taster who married a female non-taster, and they had children that were both tasters and non-tasters. Okay, there's one non-tasting child there. Okay, girl. Okay, so what does the term dominant allele mean? Okay, what does the term dominant allele mean? It means that it is always going to be expressed even when it is in the heterozygous condition. The dominant allele will always be expressed. Okay, it's always dominant over the other type of allele there. Okay, give the letter of a female in the F1 generation who is a taster. female taster. So it's only going to be, the F1 generation is MNOP. So who's the female taster? N. Okay. The genotype of individual J. Individual J must be heterozygous. Why must they be heterozygous? Because they had a daughter who is a non-taster. Okay, so the genotype of J has to be big T, small t. Okay. Use evidence from the diagram to support your answer in question 2.5.2. Okay, I just said because they have a daughter that is a non-taster, so O's genotype is small t, small t. Therefore, J has to be heterozygous dominant because the father needs to contribute a small t because the mother is small t as well. Okay, is everybody understanding that? Yes, give me a thumbs up or a yes.
Fanelo requested for you to repeat.
Okay. Okay, so if you look at the, so the question is give evidence, okay? So if you look at the offspring of J and K, you can see that you've got a female non-taster. And what is the genotype of the female non-taster? Remember, taste is dominant. Okay, so the non-taster is homozygous recessive, small t, small t. So the mother has to contribute one of those small t's, and the father has to contribute the other small t. Therefore, the father has to be big T, small t, because he is a because he is a taster. So he has one dominant allele, but he has to have that other recessive allele because he has a daughter that is homozygous recessive. Okay. Is that okay, Mfanelo?
Yes, thank you.
Okay. Okay, students, that's the last question that I have. Okay, I'm just going to go through another PowerPoint that I have. Okay, let me stop sharing. Okay. Okay, so remember, this is just to summarize some aspects of the genetic section for you. Okay, so remember, incomplete dominance, you have an intermediate. Okay, so it's a mixture. Okay, for example, a red and pink flowers, you'll get, sorry, red and white flowers, you'll get a mixture of pink. You'll get a blending of both alleles. Okay, however, in co-dominance, both alleles are neither dominant, the alleles are neither dominant nor recessive. They are both co-dominant. So both alleles will show up on the offspring. Okay, for example, a brown rooster and a white rooster, the offspring will be, a brown rooster and a white hen. Okay, the offspring will be brown and white. It won't be light brown. It won't be a mixture of white and brown. It'll be white and brown separately on the offspring. Okay, so there is co-dominance. Both alleles are equally dominant. Okay. Then our blood groups. Okay, we've got our Abo blood groups. Okay, so you have I think I have another example here somewhere. Okay, so if you look at this diagram, you can see that you've got IA and IB, which give you AB. Your IA is your A blood group, and IB is your B blood group. Okay, if you have I, small I, that is your blood group O. And this is an example of codominance, okay, because all these alleles are going to show up on the individual, okay. So if you've got IA, IB, you'll get the AB blood group. Okay, so that is how we know it's codominant, because both A and B are showing up on the individual. Okay, and the recessive is, or equally, yeah, it is a recessive allele, is small I. Okay, so that only shows when it is in the homozygous recessive form, and that gives you your O blood group. Okay. Okay. Okay, so that's a quick summary of some aspects of the genetics. Okay, so is there any questions that you might have for genetics that I can deal with? I've got some time. We can go through any questions or any other examples you want me to do. Students? Okay, let me just go through my notes and see what else I need to tell you. Okay, so when it comes to in the genetics section, okay, they can ask you some theory like about mutations and genetic disorders. Okay, so you can get different structural disorders, or what we call chromosomal aberrations. Okay. Oh, I see. Okay. S says, can you go over a question to draw a Punnett square for a dihybrid cross? Okay. Okay, let me see what I have to show you one. A dihybrid cross. Let me go back to my slide and see which one that I had. Oh, I don't have one that shows you a dihybrid. Okay, I'll tell you about it. Okay. So in a dihybrid cross, now remember you've got two characteristics. Okay. So you need to know, you'll have gametes of both characteristics. Okay. How do I get this to show on the screen quickly enough? Minisha, do you have anything on hand that we can show?
Just trying to find it. This is not
Okay,
very
yes,
clear.
me too. Okay, but okay, in the meantime, I'm going to tell you, okay? So when you have a dihybrid cross, it's going to be the same format, the same layout, okay, as what I showed you for a monohybrid. The only thing is you're going to have two characteristics, okay? So it will be, let's say you've got a tall plant and the height of a plant and the color of the seeds. Okay. Oh, sorry. Let me just see here. Okay, here's a good example. Let's use this. Okay, a dihybrid cross. Okay, so in this example, you can see that we are looking at the skin of the lemon and whether it is green or yellow. Okay, so the type of skin, smooth skin or wrinkled skin. Oh, sorry. Yeah, it's round or wrinkled. Okay, and you've got yellow or green. Okay, so those are two characteristics. So now when you're writing it, you need to put both characteristics next to each other. You must always keep the symbols representing that gene, keep it together. Okay, so always put the R's together and the Y's together. Okay, don't let it be RY. It must be RR YY. Okay, so keep each gene together when you write it. Okay, so this is how the Punnett Square is going to look. It's going to, you're going to have your gametes, which will be big R, small r, and then the other characteristic will be big Y, and small y. Okay, so now your combinations will be big R, big Y, big R, small y, small r, small y, small r, small y. Small r, big Y, sorry. Okay, so then you have to now when you're doing the cross, you have to put those two things together. So the big R will go with the big R, and then you put it together. You write it there together, okay? And then your big Y goes with the other big Y, and you write it together. So you can clearly see the two genes, okay? So that particular offspring is round, homozygous round, and homozygous yellow, okay? And the next one is big R, big Y, small y. So it's homozygous round, but heterozygous yellow, okay? It has a recessive allele, okay? So that's how you continue on with the table like that, okay? And then you have to count now. Use a highlighter, use a colored pen, okay, to count all the ones that are the same, okay? Now, as you can see, and it's something that you can just learn off, when you have two heterozygous parents, okay, so there'll be heterozygous round with yellow skin and heterozygous wrinkled with green skin. Okay, if they are both are heterozygous in a dihybrid cross, the offspring will always be in the following ratio: 9:3:3:3:1. Okay, you're always going to get that answer. Okay, if the parents are heterozygous, okay, if they are heterozygous for both traits, okay, heterozygous for both traits, you're getting 9:3:3:1 always. So that's a good way of checking now if you count it correctly. Okay, is that answering the question? You wanted us to show a Punnett square. Okay, so this is how you must write it, okay? Okay, is that fine? Do you understand that? Okay, fantastic. Okay, so I was saying that you need to also know types for genetic engineering. You need to be able to know what is cloning, okay, and what is cloning and selective breeding, okay, and we already spoke about stem cells, okay, so you need to know that. You need to know the advantages of genetically modified foods. Why do we do genetic modifications? Okay, so that we can obviously get the best of everything, the best the most favorable characteristics. Okay. Okay, and then you need to know about using mitochondrial DNA that is only found, that is inherited from the mother. Okay, and paternity testing is like using DNA from each person. Okay, and then you can identify the father in that way. Okay, and you also need to know different types of mutations and genetics disorders. Okay, so different types of mutations, you can, we have what we call chromosomal aberrations. Okay, so we get structural chromosomal aberrations like deletion, duplication, and inversion. Or we get numerical chromosomal aberrations like aneuploidy and euploidy. Okay, so obviously deletion is when we have a whole section of DNA missing or deleted. Duplication is when a whole gene is repeated. Okay. Then inversion is when a section of the DNA is reversed. Okay, that is your structural chromosomal aberrations. In your numerical chromosomal aberrations, you have euploidy, which means there's a whole another set of chromosomes. There's a total repeat of the entire set of chromosomes. Okay. Aneuploidy is when there's just one chromosome that is added on or repeated. Okay, for example, in Down syndrome, you've got a repeat of chromosome number 21. There's 2 copies of chromosome 21. So that is aneuploidy. Okay, when there's variation in the number of individual chromosomes. Okay. What else can I tell you about genetics? Does anybody have any other questions? Any other questions for me? Is there any other questions from any of the other sections that you need me to go through quickly? I've got some time. Is everybody feeling well equipped to handle the exam? Remember, the genetics section is 33 marks. Okay, it's actually the biggest section. So you must be able to do those Punnett squares and to be able to read off of pedigree diagrams and read off of like a paragraph when you are explained a situation, like when you're told the father is heterozygous and the mother is homozygous, or the father is a carrier and the mother is homozygous recessive. Okay, you in what we call like a story, you must be able to extract information from that and be able to answer questions. And they will always ask you genotype and phenotype. Okay, so you must be able to do that.
And when you get the paragraph, the best is to, in order to extract that information as you're reading, either take your pen and underline the words, or take your highlight and underline recessive or heterozygous, or is a carrier.
Yes.
Underline those words, underline which trait is dominant, which trait is negative. So when you're answering the questions, you can pull out the information much faster.
Yes. Okay, those are some tips for you in answering your genetics, but please make sure you follow those steps that I've said. Otherwise, you're going to lose marks when you're doing the Punnett squares and stuff. Okay. Any other questions? Okay, students, don't forget that you need to please register. Okay, so Dr. Singh has put up we'll put up the links there on the chat for you. Okay, if you have any further questions, you can always email us on lifesciencehelpline@ukzn.ac.za. And if you can just please click on the register form so that you can register, and we've got you on the database for the next helpline sessions that we have.
Okay, so I'm pasting the website. So I was informed that latest Monday, all recordings, all 4 sessions recordings will be uploaded onto the website. So you'll see this website is for all subjects. It's for maths, it's for chemistry, it's for life science, and for physics. So even if you only attended ours, you still have access to the other subjects, and you have this year's recordings with last year's recordings. Each are heading 2021 and 2022.
Yeah. Okay, does anybody have any further questions for us?
So if there is no questions, I'll just play the video.
Okay, we do have a short video on how you can, I think it is how you can apply to UKZN, isn't it?
Yes, let me just adjust my audio.
Part of the UKZN experience is the opportunity to be part of a community and to network with not only our world-renowned academics, but with future game changers and leaders. They do say that the extent of one's success can be attributed to those they rub shoulders with. Therefore, you must surround yourself with like-minded people. So, how do you become part of a community that is committed to your self-development? How do you become part of UKZN? First, you have to apply. All applications for undergraduate studies at UKZN are processed through the Central Applications Office, CAO. This is for both your course and student residence application. The application process is as follows. Step 1, consult the CAO handbook. This handbook provides a list of all qualifications available at UKZN. Step 2, have a look through the various courses offered and choose a program that interests you and for which you meet the minimum requirements. As one of the top 5 universities in the country, UKZN promotes academic excellence. So meeting the minimum requirements does not guarantee you a place to study. Step 3, find the program code linked to your chosen qualification. This is the code you need to enter in your application form. Okay, let me show you something. You see, a code has 3 parts. Example, for BCom in accounting, the code is KN-P-BCN or KN-W-BCN. Crazy code, right? Well, basically, the KN stands for UKZN. The middle part stands for the campus of your choice. P for Pietermaritzburg, W for Westville. Then BCN stands for the actual course. Make sure to get these codes right. Step 4, use this program code when completing your CAO form, which is available from the CAO website. Your application will be sent to UKZN. You can always check the status of your application using your CAO number, ID number, or passport number on this website, www.caocheck.ukzn.ac.za. For more information, visit www.cao.ac.za. It's that easy. Pay close attention to closing dates for qualifications. For example, MBCHB closes on the 30th of June, whilst other undergraduate programs close on the 30th of September. What are you waiting for? Apply now and shape your future today.
Okay, thank you, students, for your time, and hopefully you found all of these sessions helpful. Please watch out for our future sessions. They'll probably be around your trials time, okay? So we will be doing these sessions again for you later in the year. Okay, so thank you for your time, and I hope you found it helpful. If you have any further questions, you can email us. As I said, on Life Science Helpline at ukzn.ac.za. And we will assist you on that platform. Okay, thank you everybody and good night.